Hello, students. So, today we'll continue the chapter Microorganisms, and in this chapter, we'll study that is most important, and that is virus. Okay, and bacteria. So, virus, that is not the virus of the movie Three Idiots. Now, we are discussing here the virus like our corona that recently we have faced a lot of problems due to corona. So here the virus we'll discuss. First, let's know regarding the virus. So what is virus? Now, virus are the organism that possesses nucleic acid but lack the replicating machinery. Now, let me take the pointer. So here, the virus actually, we can say the virus or a virus, it is a submicroscopic infectious agent. Okay, because we have faced a lot of problem due to this. So infectious agent that replicates only inside the living cells. That replicates, I'm writing, you can note down this, that replicates only inside, it may be human cell, but that replicates only inside the living cell. Okay, living cell of an organism, right? So this is the virus. So virus, that means it is a submicroscopic infectious agent, and it will replicate only in the living cells of the organism. Now virus infects all life forms. Whatever the life forms you have, virus may infect, from animals to plants to microorganisms also the virus can affect. So here, that means it will affect all these things, and including bacteria, area, this they will just affect all those things. Now viruses are almost found in every ecosystem on earth, and very numerous type of biological entity also it has, and the study of the virus, that is called virology. Now what, whenever the coronavirus that affected us first time, they are researchers, those are working on the virology, they are actually doing their job so fast that we are getting the vaccines also, right? So we'll also study regarding the vaccine. First, let me tell about the virus. Now, that means now we can understand one thing, that this is a submicroscopic infectious agent that replicates when it is inside the human cell, or it is inside the microorganism living cell, or it is inside the plant also. So this is, that means it should be the virus can survive whenever it is in the living cell. So it cannot survive without a living cell. It cannot survive without any living cell. So very few examples are influenza virus, HIV, rabies virus, and polio virus, tobacco mosaic virus. So these are the different examples of the virus, and also the very new example, that is the SARS-CoV-2. SARS-CoV-2, SARS-CoV-2, COVID-2. So SARS-CoV-2, this is also SARS-CoV-2. This is also a member of the subfamily of coronavirus, or we can say that this is also a submember. This is also a member of coronavirus. Now, in this case, virus, so we can say virus are infectious agents that replicates inside a body of a host. Now, what it will do? When it will infect, first it will infect, then the host cell is often forced to rapidly produce thousands of copies. So the host cell, whatever, that means it has infected the host cell. Now the host cell, it just forced, sorry, it forced to rapidly, sorry, produce thousands of cells. Rapidly produce thousands of copies of the original virus. Okay. Now, when not inside an infected cell or in the process of infecting a cell, virus exists in the form of independent particles, and that is called virions. That is called virions. And it consisting of a genetic material, and it consisting of genetic material. Genetic material. Okay. Whenever it is independent. Now, it consists of genetic material, and that is long modules of DNA and RNA. And here, the virus, whatever, so it is the infectious agents and it applicates inside the body of the host. Whenever it will infect the host at that time, it happens that the host cell forced to rapidly produce thousands of copies of the original virus. So that's why the virus will be rapidly spread over, and so that we can see that there will be infection. Now virus is our non-cellular microscopic infectious agent, and that can only replicate inside a host cell. That means if you do not have any, it will not infect any plant, any microorganism, any human body, but any human cell. At that time it cannot do anything. Okay. Now here, the virus cannot be classified either a living organism or non-living organism, because it poses certain defining characteristics, features of the living organism as well as the non-living entities. So we cannot divide, uh, define that, we cannot do the classifications according to these things. Now, if we are classification at that time we cannot do due to this thing. Now what happens, how it will work, we'll see later on. So virus, that is a non-cellular infectious entity and made up of genetic material and protein, and that can invade and reproduce only within the living cells of bacteria, plant, animals, and this several things. Now, here, one thing you have studied, that is the airborne transmission, right? Some virus, that is airborne transmission. So, airborne transmission means it is defined as the spread of infectious. This virus will spread, and that means that it is caused by the dissemination of the droplet nuclei, and that remains infectious when suspended in air. That is the airborne transmission. Sometimes we are calling that virus is also airborne transmission. It is also may spread over due to the airborne transmission. So the virus is an infectious microbe consisting of a segment of nucleic acid, either DNA or RNA, and surrounded by a protein coat. Surrounded by a protein coat. So it consists of It consists of a segment of nucleic acid, either DNA or RNA, and which is surrounded by a protein coat. Surrounded by a protein coat. Okay. So this is the virus. Now, next is for virus, it cannot replicate itself. We know that, and it requires some host cell. So the host cell will be infected, and this host cell will replicate the different thousands of the this will replicate thousands of the viruses. Now, this is because the virus lack the required cellular machinery. It does not have the cellular machinery. That's why it cannot replicate itself. So it requires some cell. Now it enters and attaches itself to the specific host cell and injects its genetic material. It injects its genetic material and reproduces by using the host genetic material, and finally the host cell splits open and releasing the new virus. So this is the process how the virus will be spreaded. So what it will do, it enters and then it attaches itself to a specific host cell and injects its genetic material and it produces by using the host genetic material, and finally what happens, the host cell will splits open and it releases the new viruses. Okay, so virus can also be crystallized, which no other living organism can do. It also we can be crystallized, and it is this factor that lead to the virus being classified in the gray area between living and the non-living, because it can be, this can also be crystallized. That's why it is, we have told that it can be classified in the gray area, that means in between living and non-living things. Now the structure and function of the virus. Okay, structure and function of the virus. So viruses, these are very tiny. These are very tiny, and this coronavirus, we have seen a lot of diagram of this coronavirus. These are the this type of diagram we have drawn, but this is not the biological diagram. Now viruses, these are tiny and smaller in size and ranging from 30 to 50 nanometer. 30 to 50 nanometer, and you please remember 1 nanometer, that means 10 to the power minus 9 meter. Okay, so 30 to 50 nanometer, and they do not have a cell wall, but surrounded by, they do not have any cell wall. They do not have a cell wall, but what it has, that is surrounded by a protective protein coating that called the capsid, and it is surrounded by capsid. Okay, and it is surrounded by the protective protein coating, and that's why it is the, and this protective coating of the protein coating, that is called the capsid, and it can be seen as a genetic element and is characterized by the combined evolution of virus and the host. They contain either DNA or RNA as the genetic material, as the genetic material. So here the DNA, that is the deoxyribonucleic acid, and this you know that this they contain either RNA or DNA as genetic material. Okay. Now this is one structure we have seen. Now first, this is the capsid. This one is the capsid, and this one is the DNA. And here you can see this is the capsid. This is the capsid. This one is your RNA, and this one is your enzyme. This one is the envelope protein. We have told one protein coating is there, and this one is the envelope. Okay, this one is the total envelope, and this is the envelope protein. Here you can see these are the like birds type, and these are the envelope protein, and these are the enzymes, and this is the RNA. This one is the RNA. So this is the RNA. This is the RNA. And here you can see this is the this point, this every these are the envelope protein, and this one is the envelope, and this is called capsid. This one is called capsid, the coating of protective coating. This coating is called the capsid. This coating is called the capsid. Okay, this coating is called the capsid. And here this is the structure, okay, that we have discussed earlier. Now, viruses mainly depend on a host to deliver the complex metabolic machinery of prokaryotic or eukaryotic cells. So, they require one prokaryotic or eukaryotic cell for the their reproduction, and they deliver the complex metabolic machinery. And the main task of the virus is to carry the what DNA and RNA genome to the host cell, which then can be transcribed by the host cell. So here you can see that this is the one structure of the virus. Here you have the nucleus, and this is the nucleus, and here you have the lysosome. This is the lysosome. Ribosome also. This is the ribosome. This yellow part. This is the ribosome, and here you have the cytoplasm also. You have the cytoplasm also, and cell membrane also you have here, and DNA you have here the DNA, and you have some mitochondria. Here you have some mitochondria. Here you can see the mitochondria. So the main task of the virus, that is to carry the DNA or RNA genome to the host cell and to the host cell. Okay. The viral genome is packed in a capsulated symmetric protein. The viral genome that is packed in a capsulated symmetric protein. And this protein associated with nucleic acid, that is called a nucleoprotein. And it produces the nucleocapsid with the genome. This will create the produces the nucleocapsid with the genome. Okay. So here what we have studied, that viruses, they totally dependent on the host, and to deliver the complex metabolic machinery, and they will just this virus, the main task, main job is that the virus is to carry the DNA or RNA genome to the host cell, and this viral genome will be packed in a capsulated symmetric protein, and the protein associated with the nucleic acid. So here you can see one structure of the virus. So that this protein, as whatever associated with the nucleic acid, that is called a nucleoprotein. That is called nucleoprotein, and it produces nucleocapsid with the genome. It produces nucleocapsid with the genome. Okay. Now the characteristics of the virus. Virus, it does virus lack the cellular organelles and cytoplasm. They fail to perform any metabolic activities. If they are infecting any cell, that means any cell of the living organism, then only they can create these metabolic activities. Most of the virus, they contain RNA or DNA, but not both. But not both. Either DNA, they have either DNA or RNA, both all the viruses. And virus reproduce at a tremendous pace. That means very rapidly it will just spread out. Very rapidly it will spread out. They make use of metabolic machinery of the host cells, and virus cannot grow and divide. Virus cannot grow and divide. And they produce and assemble new viral components inside the infected host cell. If it is outside, at that time it has no strength, but whenever it is inside the cell, at that time you can see that it can produce and assemble new viral components also whenever it is inside the cell. Okay, it may be the host cell, maybe of the plant, maybe of microorganism, maybe of the human also. Okay. Now, the bacteriophage or HIV. These microbes belong to the family Viridae and genus virus. And virus could not be placed, virus could not be placed in any of the kingdom because they are practically neither living nor dead. That's why, because whenever it is outside of the host, you can see that it will just act as non-living things. But whenever it is inside any host cell, at that time you can see that it will act as the living things. So the term virus that was coined by whom? Dutch microbiologist Martinus Willem. So in the year 1897, and it is derived from the Latin word which means poison or venomous substance. Poison or venomous substance. So the bacteriophage or the HIV and HIV, these are the belongs to the family Viridae and the genus virus. And this has been this virus term. The meaning of this term, this is a Latin word, and the main term is the meaning of this term is the poison, and this has been invented by Dutch microbiologist Martinus William. Okay. Now, once a susceptible cell is infected, a virus starts the cell machinery to generate the more virus. So, already that means it requires the cell machinery. It can do only if it is it will infect any cell. Until unless it cannot do anything. And viruses are composed of DNA or RNA, and the size that is ranges from 20 nanometer to 250 nanometer. 20 nanometer to 250 to 250 nanometer. Can you see the 20 nanometer? That means your 20 into 10 to the power minus 9 meter. How much tiny is there? Okay, so therefore they can only be seen with an electron microscope. We cannot see the virus here and there with our naked eye. We need the microscope. So we have already in the first slide, we have already told that that is the submicroscopic this agent. This is submicroscopic agent. Okay. Now, many viruses have either DNA or RNA as the genetic element and the nucleic acid with single or double strands. And the nucleic acid with single or double strands. And the whole infectious virus that is called viral has nucleic acid and outer shell of the proteins. That you have seen there is a protein cover here. So you have seen also there is a protein cover around the there will be a protein cover around this virus. So it has a protein cover, and this simplest virus includes DNA or RNA for encoding 4 proteins, and the most complex encodes 100 to 200 proteins. 100 to 200 proteins. So here different properties you have in the virus. Now they are non-cellular organism. This is number 1. And this non-cellular organism that is enclosed with the protective envelope that we have seen in the picture. We have seen in the picture the protective envelope. Already we have seen in the this there will be some this type of protective envelope we have. This type of protective envelope we have. This is the like this you have. I am just drawing very roughly. So this type of protective envelope you have. Protective envelope you have. Okay. And in this case, the presence of spikes helps in attaching the viruses to the host cell. It has some spikes also, and that will help to attach it to the another cell. These viruses do not grow, neither respire nor metabolize, but they reproduce. But whenever one condition is there, whenever they are inside any host cell, at that time only they can reproduce. The virus that do not grow, neither respire nor metabolize, but they will reproduce. And they are surrounded by a protein coat that is called a capsid and have a nucleic acid core comprising of DNA and RNA. DNA and RNA. DNA and RNA. Now they are considered as both living, non-living, as already I have told this one. These viruses are inactive when they are present outside the host cell. But whenever inside the host cell, you can see that whenever inside the host cell at that time they are living. They are living organisms. Now these viruses that cause several infections and it will reproduce within the host cell by using the enzymes and raw materials, but it requires the host cell also. Cell. Now, classification of the viruses. The virus can be classified primarily on the phenotypic characteristics, core content, chemical composition, capsid structure, size, shape, genome structure, modes of replication. According to that, we can classify this thing. The Baltimore classification is the most commonly classification, and it has been developed by American biologist David Baltimore in 1970s, and he was awarded the Nobel Prize for his invention. Now, classification based on the presence of nucleic acid. Presence of nucleic acid. Now, first is the DNA virus. Let me take the highlighter first. So first is the DNA virus. Now the virus having the DNA as a genetic material, we know that the virus either it can have DNA or RNA. So DNA virus, that means it having the virus that having the DNA as its genetic material, and there are two different types of DNA virus. One is single stranded DNA virus like parvivora virus and and also picornaviruses. Double-stranded virus, DNA virus also we have. And next is the RNA virus. DNA virus and RNA virus. Let's study about RNA virus. So here the RNA virus here, the virus it has the RNA as the genetic material. RNA as the genetic material, and it also has two different types, and one is the double stranded RNA virus and another is the single stranded RNA virus. And this poliovirus, hepatitis A, rabies virus, influenza virus, these are the examples of single stranded RNA virus. Single stranded RNA virus. And this single stranded RNA virus, it is again classified as the two positive sense RNA and negative sense RNA. So it has another two classification, positive sense RNA and negative sense RNA, right? So first what we have studied, that we have studied DNA virus. We have studied DNA virus, and next we have studied RNA virus. In DNA virus, it has the genetic material that is DNA, but in RNA virus, it has the genetic material that is RNA. And both have the classification also. There are two different types of DNA virus, that is the double-stranded DNA virus and single-stranded DNA virus, as well as for the RNA virus also, it has the classification, and there are two different types of RNA virus. Two types of RNA virus. One is the double-stranded RNA virus, another is the single-stranded RNA virus. And this single-stranded RNA virus, it is also maybe of two types. One is the positive sense RNA, another is the negative sense RNA. Another is the negative sense RNA. Now the classification based on the structure or symmetry. Structure or symmetry. Here the complex virus. First thing is the complex virus, that pox virus. This is the complex virus. Radial symmetry virus. That is the bacteriophage. Radial symmetry virus. Cubical symmetry shaped virus. Cubical symmetry shaped virus. That is there are several viruses like this. Rod or spiral shaped virus or helical symmetry virus. So we have complex virus, we have radial symmetry virus, we can have cubical virus, you can have rod shaped virus. Next, classification based on the replication property and site of replication. Replication property and site of replication. Here, virus invade into the host cell where it will replicate and assembly within the cell organelles. So, replication within the cytoplasm of the host cell and replication within the nucleus and the cytoplasm of the host cell. So, here you can see that there may be of two types. One is the replication within cytoplasm, of the host cell, Another is what? Within what? Our within our nucleus and cytoplasm of the host cell, like influenza virus, pox virus. These are the they will replicate within the nucleus and cytoplasm of the host cell. Now, replication within nucleus. Okay, there is no cytoplasm, so all DNA virus except pox virus, these will replicate within the nucleus of the host cell. Replication of the virus through the double-stranded DNA intermediate. All DNA virus, retrovirus, these are the examples. Replication of the virus through a single-stranded RNA intermediate. So all RNA virus except retrovirus, and tumor causing the RNA viruses. Okay. And classification based on the host range. Now, there may be different types of viruses according to that based on the host. First host is the animal virus. Now, the virus infect by invading the cell of the animals and then including the humans, and prominent example of this animal virus includes the influenza virus, mumps, rabies virus, polio virus, herpes virus. There are different types of viruses. Now the plant virus also. Here, these viruses infect plants by invading the plant cells, and well-known example is the potato virus, tobacco mosaic virus, beet yellow virus, turnip yellow virus, cauliflower mosaic virus. These are the different plant viruses. Bacteriophage, this virus which infects bacterial cells known as bacteriophage. Now, like such as MV11, RNA virus, lambda phage. So there are different type of bacteriophage, this type of virus also. Then insect virus. So insect virus, the virus which infect the insects, is known as the insect virus. So it will just infect the insects. So this is the insect virus, and it is all called also viral pathogen of insects. It is also called viral pathogen of insects. Viral pathogen of insects. And these viruses are most powerful viruses, and these viruses examples are the different examples are there, like entomopox virus. These are the different types of insect virus. Now, classification based on the mode of transmission, that means how they are infecting. First is the airborne infections, like swine flu, rhinovirus. These are the airborne infections. Fecal oral route. Here, hepatitis A virus, poliovirus, rotavirus. So, sexually transmitted disease, like transmission of the virus through the sexual contacts with the infected person. Retrovirus, human papillomavirus. So, there are different types of sexual transmitted disease also. And this type of virus also, airborne infections also due to fecal route, oral route, and transfusion transmitted infections. Transmission of this virus to the blood transfusion, like hepatitis B virus. Here, hepatitis B virus, transfusion transmitted infections. Then zoonosis. Zoonosis, this transmission of the virus through the biting of infected animal or birds or insects. It can happen by biting of infected animals, birds or insects to human. And rabies virus, like rabies virus, then alpha virus, you have Ebola virus. So these are the zoonosis. Okay. Now, classification based on the structure: complex virus, radial symmetry, cuboidal rod. Already we know these things. Next is the structure of the symmetry, that is complex virus, radial symmetry, cuboidal rod. Please remember this thing. Now, list of viral diseases. So many viral diseases are there. AIDS, acquired immunodeficiency syndrome, Ebola, influenza, SARS, this is very important, and chikungunya, smallpox, these are related to different virus disease. Now, the main difference between the virus and bacteria. Bacteria, in this case, outer cell wall, according to the outer cell wall, bacterial cell wall is made up of peptidoglycan, and viruses do not contain a cell wall. But in case of bacteria, cell wall is made up of peptidoglycan. And the genetic material in the virus, that is enveloped by a protein coat, that is called capsid. And bacteria, these are large in size, 900 to 1000 nanometer. But in case of virus, what we have studied, it is 20 nanometer to 200 nanometer it may be. The size range from 30 to 50 nanometer, but it may go up to 200 nanometer also, but not like that 900 to 1000 nanometer. So it is very tiny. Then with respect to the bacteria. Now, non-living living, they are living organism, and there is a gray area here within the host at that time living. Now, mode of reproduction asexually by binary fission, the bacteria. And here reproduction, it will insert their genome in the host genome, then make multiple copies. Host dependence, that host independent production, and host dependent reproduction. There, for bacteria, the host cell is not required here, but here you require the host cell. And ribosome are present in bacteria while it is absent in virus. This difference is very important. Several points I am adding. So in this way, you have to write in your examination also. Now, RNA, DNA or RNA is present in the cytoplasm, and virus, DNA or RNA are enveloped inside the protein coated capsid. Infections, generally localized infection, and here generally systematic, like flu. Okay. And disease like pneumonia, meningitis, food poisoning, this type of bacterial disease. Virus disease, common cold, polio, smallpox, hepatitis, AIDS, these are the treatment. Antibiotics for bacteria, virus, vaccines, and antiviral drugs. And examples are that there are so many examples of the bacteria like Salmonella typhi, and here you have the coronavirus, right? That just suffered, we have suffered a lot of uh, almost 2 years due to this coronavirus. Now, what is bacteria? So, bacteria, already we know that this is a prokaryotic microorganism. That means it does not have any cell membrane, and they are found everywhere, and they can survive in any conditions, any harsh condition, in the hot spring also, in the deep ocean also, snow also, they can survive everywhere. And disease causing by the bacteria are known as the pathogens. This is the main bacteria here. You can see the structures. These are the pillars, and this is the capsule where you have the cell wall and plasma membrane here, and nucleus, that is DNA, ribosomes present, cytoplasm here, flagellum. With this, they are just locomotion. They are doing their locomotion. And characteristics of the bacteria, it is unicellular and some of the bacteria, they are multicellular, and bacterial cells lacks membrane bound organelle, and bacteria cell has ribosomes where protein assembly takes place, and bacteria play a very important role in the ecosystem. Bacteria, that is not only very harmful for us, it is also helpful for us, because for the medical purpose and agricultural industry we are using the bacteria also. Thank you.